Good morning, everyone. Thank you for attending the Buki Cannabis Summit webinar series. Today we will be covering the evaporation portion of this series, and the rest of the topics will continue throughout the rest of the week. This is today's agenda. I will keep the presentation short and focused on the listed topics. The purpose of this webinar is to hopefully increase your current knowledge on rotary evaporation and provide you with tips on how to optimize your process. First, we'll get into winterization and how rotary evaporation fits into that process. And then we'll move on to the theory. Understanding the theory gives you a better handle on how to properly optimize your specific process. And then after understanding that, we will also move into solvent recycling and how solvent recycling can greatly benefit any process downstream. So going into the introduction, I'm your presenter, Donald Maybell III. I am the current product specialist here at Buki. I handle laboratory, industrial, and parallel evaporation, as well as spray drying, freeze drying, and our chromatography line. Our first topic, winterization. What is the winterization process? It is the process of removing a good portion of the crude material from your cannabis extract. This is done by freezing the extracted material in a laboratory freezer to separate the layers. Once the layers are separated, you gravity filtrate the desired layer and continue on to the next step of the process, rotary evaporation. Rotary evaporation comes into play by concentrating and pulling off the solvent so you are just left with your concentrated oil. After you've concentrated your oil, then the next thing is your post-processing, whatever that may be. You might need to further purify it, and that's where chromatography will come into play. You can get more information on that later this week. Rotary evaporation, or our rotovapor. Reasons to use a rotovapor are to concentrate samples, for solvent recycling, to run reflux reactions, depending on the glassware configuration that you have, and to separate mixtures of solvents. And you can do this both at the laboratory bench scale and at the industrial scale. Now we get into the working theory of rotary evaporation. Rotary evaporation is a distillation process, and for any distillation process to begin, there must be some form of energy. That form of energy is usually in the form of a heating bath. Usually how it goes is you have the energy from the heating coils goes to your heating medium, which is usually water or a high temperature fluid, and then from the high temperature fluid, the energy transfers from there into your sample. The next part of the process is your vacuum. Since this is a vacuum distillation process, you pull down the vacuum to control the boiling point. By doing this, you can keep the sample at a reasonable temperature to protect your sample, and you get a nice efficient distillation process which gives you higher throughput and allows you complete control over the entire process. And then last would be your condensation. How much energy you put into the system, you must take out as well. Distillation is a two-part process. It consists of evaporation and condensation. If you don't have good adequate condensation, what can happen is any vapors that get past the condenser will go straight into your vacuum source. And then at the bottom of the condenser is usually a receiving flask. So depending on what you're actually doing with the condensate that condenses down into your receiving flask, you can either take this processed solvent and dispose of it properly, or if you're solvent recycling, you can reuse it throughout your workflow. So these are all the parts of the main system. And it's very important to understand this so that when you go to optimize your system, you know which part of your process to actually pay attention to. Distillation under vacuum. We distill under vacuum because pulling a vacuum on the system controls the boiling point of the solvent. So let's first understand that evaporation only happens at the surface of a liquid. Because of that atmospheric pressure, you have more particles pushing down on the surface of that liquid. So that means you have to add more energy to the liquid in order to really get it to go from that liquid phase to the vapor phase. When we pull a vacuum on the system, it pulls particles out of the system. Since we pull those particles out of the system, that means there's less particles pushing on the surface of the liquid. So not as much energy is needed to get the liquid from the liquid phase to the vapor phase, i.e. the lower boiling point. And because we are working at a lower boiling point when we pull a vacuum, that means when we add extra energy to the system, Because the liquid is more ready to go from the liquid phase into the vapor phase, you can increase the throughput or the flow of particles from the liquid phase to the vapor phase. So by adding that extra energy and pulling the vacuum on the system, you can greatly optimize your process. Since we are distilling under vacuum, it's very important to understand the risks of bumping. Bumping is when you have an explosive nature of the sample that you're working with, and you have solvent and your sample splashing around or almost exploding all over the entire system, whether it be the flask, and then it could possibly go up into the condenser area as well, which would mean sample loss. So how this usually happens are one of two reasons. Usually you overheat your liquid, so you have temperature gradients, so you're not mixing fast enough. So temperature gradients, you get cold solvent mixing with hot solvent. They mix very rapidly because there's such a large temperature difference. You get basically slow convection current inside, and you get the hot solvent on the bottom exploding to the top and then exploding all over your system. The next is if you pull the vacuum too fast. If you pull the vacuum too fast, then what happens is air bubbles trapped inside of the actual solvent rip towards the top, hit the surface, and then fly out of the system very, very quickly, which also sends your sample flying all over the place. Air bubbles tearing the portion of the liquid off is one of the main causes, and as I mentioned before, is because you pull vacuum too fast. So it's important that you reduce the bumping by either rotating. So we rotate the flask in rotary evaporation, and then also pulling a slower vacuum on your system. So don't pull down to the set vacuum all at once, do it gradually. So now that we understand distilling under a vacuum and the importance of not bumping your sample inside of the system, we start to get into the optimization. What parameters do you need to have to get good, efficient evaporation without hurting your sample or providing sample loss by bumping? So we have the delta 20 rule. So we usually say you want a 20-degree difference between your heating temperature and your vapor temperature or the boiling point, and then another 20-degree difference between that boiling point and your condenser temperature. So just a quick example, 20, 40, 60 is always a good starting point. So you want 20 for your cooling, 40 for the target boiling point, and then 60 on the actual heating bath itself. And what this does is it provides that good energy transfer. How much energy you put into the system, it gets taken out in an efficient manner as well. And then it also gives you a decent rate, so you don't have to add a whole lot of heat to the sample to overheat it, but you get good distillation rates because the solvent comes over in a nice uniform manner. So pressure needs to be adjusted, so depending on the solvent that you're working with. You'll need to make sure that you bring the pressure down enough so that the vapor temperature is actually at 40 degrees Celsius. Process optimization. So now we get into how to actually optimize your specific process. So just a quick example, I told you 60, 40, 20. So that means if you're working with ethanol, you will want your pressure to be 175 millibar. Now, if let's say for some reason 60-degree temperature on the heating bath is too high for your process, that is not a problem. You adjust the pressure down lower, but you also have to adjust your heat. So you adjust it down to 40 degrees C. That means your target boiling point at 77 millibar is 25 degrees C. Now you have a cooling temperature of 5 degrees Celsius. Now these are two very good examples, especially to get your process started. Depending on how powerful your chiller is, you can go lower on the cooling temperature, but I'll get into that a little bit later. But in the case that you want to optimize outside of the Delta 20 rule, that is also possible. But it's very important to pay attention to the cooling capacity of your condenser, and then make sure not to bump the sample into the condenser area and all over your flask. So important recommendations, as I mentioned previously, make sure to ramp down the pressure instead of pulling down extremely quickly. Make sure you have adequate cooling capacity. And then the last little bit is making sure you have the proper flask size. As I mentioned, evaporation only happens at the surface of a liquid. So maximizing the surface area of the flask that you're working with is extremely important. And then also have to think about retrieving your sample after the process has finished. So we use a beaker flask. We offer a beaker flask. You can twist off the top and then fit your whole hand into the actual flask. If you're using a traditional flask that has a 2440 joint or a 2932 joint, it can be a little difficult trying to scrape 2 kilograms of product out of a flask of that size. So just be aware. So continuing on with the process optimization, the cooling capacity. So this cooling capacity value that I've been talking about is listed in any technical data sheet for any chiller that you look at. So here's an example of our benchtop chillers. So we have the F305, the F308, and the F314. So their cooling capacities at 15 degrees Celsius are 550, 900, and 1400 watts. So this is very important because the temperature that's listed and then the cooling capacities at that temperature are going to tell you how much energy can be taken out and the chiller can hold constant at that temperature. So most people don't know, but as you go to a lower temperature, the cooling capacity actually decreases. That's because the chiller has to work harder to hold that temperature. So what will happen if you don't have adequate cooling capacity is you will see the chiller heat up over time. So an example, if you set the chiller to 5 degrees Celsius and it does not have adequate cooling capacity, what you will see is throughout the process, the chiller will heat up from 5 degrees Celsius to 10 degrees Celsius. So that means that at 10 degrees Celsius, you do have adequate cooling capacity, but at 5 degrees, you do not. So if you want to keep the temperature constant at 5 degrees, that means you're going to need a larger chiller. So that's when you would upgrade. The other thing to keep in mind is how big the condenser is that you're working with. So it's very important to pay attention to the condensation level on the coils as you're condensing the solvent. If the condensation level is higher than 3/4 up the condenser, that means that some of those vapors are probably escaping into your vacuum source. So you need a stronger chiller, or you need to tone down your parameters. So you're distilling too fast for the system to handle. So it's very important that throughout your process, or even before you buy a unit, make sure you have adequate cooling capacity so that you can get the highest throughput and not halt your process or hurt your throughput by not having a large enough cooling capacity. So all the pictures that I've told you about optimizing your process have been on the benchtop rotary evaporator. But what about industrial? Easily enough, everything that I've told you applies not only to the benchtop version of the process, but also the industrial part. It's very important that you maximize your throughput, optimize your settings, and actually the settings on the benchtop unit directly apply to the industrial scale. You're just working with larger volumes. So that means you're going to need a larger flask. For one of our standard units, that will be a 20-liter flask. To maximize your throughput, make sure you fill the flask up to at least halfway. But also be aware of weight limits, because obviously you're working with glass. You don't want to break the glassware. But the Delta 20 rule still applies. So you can use the Delta 20 rule as a good starting point. If you are ready to go outside of that starting point, you can definitely optimize further, maybe increase the heat a little bit, or drop the chiller down if you're not getting fast enough condensation. But do be aware, especially if you're working with mixtures of solvents, that after you pull one solvent off, you either may need to drain the receiving flask to get the second solvent to come off, because if you pull the first solvent off and then you drop the pressure even more, what can happen is you can get reboiling. If you get reboiling in the receiving flask, then you can easily overload the condenser. So just food for thought. But in the industrial scale, everything still applies. So the best way to optimize the process is by automating your process. So to prevent bumping and foaming, the bumping part, having good vacuum control is extremely important. Proper vacuum control not only gives you a target boiling point, but it's the best way to have an efficient distillation process. Why? Because with a vacuum controller, you can optimize your process and get reproducible results. Because the vacuum controller will not only drop the vacuum down to the exact value every time, but it can help regulate the vacuum as well, especially if you're going outside of optimized settings, outside of the starting settings to better optimize your process. A controller is the best way to experiment with what settings do and don't work with your process, and then how to properly control those so that you have the same exact settings from run to run. And then also protecting your vacuum source. If you don't have proper vacuum control, what can happen is if the vacuum, let's say you're doing it manually, you open up the inlet valve on the back of the unit and let air into the system to aerate, you can actually blow sample all over the flask. So now instead of your concentrated sample remaining in the bottom of the flask, now you have to scrape it off the walls of the flask, which can be both time consuming, and in the event that you aerate way too quickly, it can actually blow sample up into the condenser area. And then there's also the fact of, providing enough heat, having good rotation, all of that stuff. And then just controlling the vacuum level pretty accurately. If you can try to control the vacuum level by hand, especially without any kind of vacuum gauge or a way to read the vacuum, you can easily bump the sample by pulling the vacuum too hard, too quickly. Now, onto the foaming part. Here at Buki, we have a patented foam sensor, both on the benchtop and the industrial scale. What the foam sensor does is it detects that the sample is foaming, or even for slight bumping situations. So if your sample starts to bump, the sensor will detect it and it will automatically aerate the system. By doing this, your sample stays protected and someone doesn't have to sit there and watch the process run for however long it takes, especially if you're working with larger volumes. Each run can take between 45 minutes up to 2 hours, depending on what you're working with. So in the event that you are doing a process that is that time consuming, it's more efficient to have everything automated by a controller and a foam sensor, as opposed to having a user there just standing there and watching the process the entire time. The next step in optimizing your process is making sure to look at the adequate accessories that fit your specific application. For example, there are amberized flasks available. Using the amberized flask, especially if you have a photosensitive material, is a good way to protect your material throughout the entirety of the process. And then another is the flask handler. This is more for the industrial scale, but when you move to industrial scale processes, since you're working with larger volumes, that means you have larger vessels as well. Since the vessels are larger and they're all glass, the main thing you want to do is avoid droppages, which can lead to massive sample losses. And not only that, but it can harm the user. So making sure you have small things like a flask handler not only protect the user, but it protects the sample, it protects the expensive glassware, and our flask handler actually doubles as a base for the flask. So not only can you use it to move your flask around the lab, but when you actually go to store empty flasks, you can use the flask handlers for that too. So those are just a couple of accessories that are available on the market and in our portfolio. But making sure to look through and look at all the accessories that are available and things that might be of interest to your specific application. So us here at Buki, we have multiple solutions, especially for cannabis extract applications. The main one being our descending or double descending R220 Pro. So as you can see listed here, not only does it provide a large cooling surface area so you get adequate cooling capacity, but we also have high heating capacity so that you can really get that high throughput that you need. On top of that, we do pay attention to regulations. So we do have CSA and UL certification so that you can fit these solutions inside of any laboratory environment unless you're going to explosion. The next thing is the versatility with many different applications. Because the cannabis market is advancing and growing very quickly, the processes might change over time as well, but luckily enough, the R220 Pro works for various different applications. So in the event that conditions do change and you're working with different solvents, that's not a worry because this can tackle almost anything. Next is protecting your sample. One of the good things, like I mentioned before, that optional foam sensor. We've designed the system to minimize overloading and bumping, so proper vapor flow through on the instrument. A good flow path of the vapors gives you very, very good condensation and just really maximizes that throughput while still protecting your sample. And then a very big thing is the integrated control. Integrated control means very precise control. So when you set something, it's going to stay at that set point and is not going to deviate from that set point unless you program it to. That increased automation comes in parallel with the integrated communication. The interface offers single button start. So once you have your method figured out, literally all you have to do is walk up to the system, press start, and it will run the exact same every single time. And then the methods function, in the event that you don't want to just go down to a specific value and then hold at that value, you can actually have it do slow ramps. You can have it drop down to a specific vacuum pressure. And then after a certain amount of time, you can have it drop to an even lower pressure, especially towards the end of the process where you're trying to get that last little bit of solvent out of your sample. That's where the methods function is very beneficial. And then also there's the safety aspect. Along with the certifications that I mentioned previously, the pump that we offer is a diaphragm pump, so it's chemically resistant. Your solvent, if solvent were to get into the pump source, then it would only come in contact with the PTFE membrane or the glass front on the front of the vacuum pump. We made the front of the vacuum pump glass so you can see when the vacuum pump is actually getting dirty, so you can properly clean it before any harm does come to your pump. We also do have overheat protection on the bath and then an optional secondary condenser on the back of the vacuum pump. In the event that a little bit of solvent does overload the condenser and go into your vacuum source, you want to make sure that you catch it on the back end with the secondary condenser so that the user is not breathing in those harmful vapors. The next thing I want to go over is the continuous operation. So we do have a continuous model RotoVapor. It's the R220 Pro Continuous. And this is more tailored for solvent recycling. And the difference between this and a regular unit is you have a number of valves that allow the sample to the receiving flask to automatically drain, and it allows you to automatically fill the evaporation flask. Completely automated process for solvent recycling so that especially after post chromatography, you can clean up all that solvent and you can possibly reuse it for extraction or other processes that you have within your workflow. So just a quick understanding of how solvent recycling works and how the automated process works. This is a good picture or a good animation to show you how that process goes about. So essentially you have all of your waste solvent. It would automatically fill the evaporation flask, which is the yellow liquid there. It fills up to the level sensor. Level sensor gets tripped, and then it stops filling it. And then the evaporation process occurs. That's where you see the blue going. So that's the solvent condensing down into the receiving flask. There's a level sensor in the receiving flask. It fills up, the level sensor gets tripped, and then you see the receiving flask being automatically drained via the 3 valves on that as well. Gives you a complete automated process and allows you to process all that solvent with very, very minimal user interaction. So this allows you to reuse solvent for future extractions, as I mentioned before, and you get to save on disposal costs, save on solvent purchase costs, and it also remains environmentally friendly. As I mentioned before, having that proper condensation, so that means the minimum amount of solvents are going into the atmosphere, and you just have that automatic distillation with the R220 Pro Continuous. That concludes this presentation. Thank you all for attending. If you do have any questions, please reach out to us. You can call into our home office or submit some questions via our website. Thank you very much.